2024 Copyright AP Biology Simplified, all rights reserved. Welcome to AP Biology Unit 3, Cellular Energetics. We will be covering 7 topics in this unit. 1. Enzyme structure. 2. Enzyme catalysis. 3. Environmental impacts on enzyme function. 4. Cellular energy. 5. Photosynthesis. 6. Cellular respiration. And 7. Fitness. These are the 7 topics, as delineated by College Board, for Unit 3 of the AP Biology exam. I will combine certain subunits when reviewing for simplicity. Let's get started. Topics 3.1 to 3.3. Enzyme structure, enzyme catalysis and environmental impacts on enzyme function. By the end of this lesson, you will understand the structure of enzymes, their role in accelerating biological reactions, and how environmental factors influence enzyme function. Enzymes are biological molecules that act as catalysts, speeding up chemical reactions in living organisms. They are essential for various physiological processes. In this lesson, we will explore the structure of enzymes, their impact on the rate of biological reactions, and how environmental factors influence their function. There are 4 parts in this lesson. First, enzyme structure. Enzymes are composed of one or more protein chains, folded into specific shapes. These shapes are crucial for their functions, as they determine the enzyme's active site and, consequently, their substrate specificity. The active site is a pocket or crevice on the enzyme surface where the substrate binds. The active site has a specific shape and chemical properties that complement those of the substrate. This complementary fit ensures that only specific substrates can bind to specific enzymes. Second, mechanism of enzyme action. Enzymes facilitate chemical reactions by stabilizing the transition state of the reaction. They achieve this by providing an alternative reaction pathway with a lower activation energy. Lower activation energy means that fewer collisions between molecules are needed for the reaction to occur, making the reaction faster. When a substrate enters the enzyme's active site, it forms an enzyme-substrate complex. In this complex, the enzyme stabilizes the transition state, allowing the reaction to proceed more easily. After the reaction, the products are released, and the enzyme is free to catalyze more reactions. Third, factors affecting enzyme activity. Enzymes have an optimal temperature at which they function best. At temperatures too high or too low, enzymes can denature, losing their shape and activity. Extreme heat disrupts the enzyme structure, while extreme cold slows down the movement of molecules, affecting the collision frequency. Enzymes have an optimal pH at which they are most active. Deviations from this pH can alter the enzyme's shape and disrupt the interactions between the active site and the substrate, affecting the enzyme's function. Initially, increasing the substrate concentration increases the rate of the reaction because more collisions between enzymes and substrates occur. However, there is a limit. Once all enzyme active sites are occupied, adding more substrate doesn't increase the reaction rate further. Increasing the enzyme concentration generally increases the rate of reaction because there are more active sites available to bind with substrates. This continues until the substrates become the limiting factor. Fourth, environmental impacts on enzyme function. Enzymes from organisms living in extreme environments, such as hot springs or polar regions, have adapted to function optimally at high or low temperatures. These extremophiles have enzymes with structures stable under extreme conditions. Enzymes in different parts of the body have adapted to function in specific pH environments. For instance, enzymes in the stomach work best in highly acidic conditions, aiding in digestion. Certain chemicals, including heavy metals and specific drugs, can affect enzyme activity. Some chemicals act as enzyme inhibitors, binding to the enzyme and preventing it from interacting with the substrate. Others can activate enzymes or alter their conformation. Enzymes are regulated by various factors within living organisms. Hormones, cellular signals, and other enzymes can activate or inhibit specific enzymes, regulating metabolic pathways and ensuring proper physiological responses. Common misconceptions about enzyme structure, catalysis, and function. Misconception number 1. Enzymes are consumed or used up during chemical reactions. Correction. Enzymes are not consumed in the reactions they catalyze. They remain unchanged after the reaction and can be used repeatedly for multiple reactions. Misconception number 2. Enzymes provide energy for reactions. Correction. Enzymes do not provide energy for reactions. Instead, they lower the activation energy required for a reaction to occur. By lowering the activation energy, enzymes speed up reactions but do not provide the energy needed for the reaction itself. Misconception number 3. Enzymes change the equilibrium of a reaction. Correction. Enzymes do not alter the equilibrium constant of a reaction. They facilitate reactions in both the forward and reverse directions, allowing reactions to reach equilibrium more quickly, but not changing the equilibrium position. Misconception number 4. Enzymes work for specific reactions only. Correction. Enzymes are highly specific for their substrates, but some enzymes can catalyze multiple reactions. Their specificity arises from the complementary shape of the enzyme's active site and the substrate, allowing a specific reaction to occur. Keep in mind that the equation for Gibbs free energy is beyond the scope of the AP exam, according to the College Board AP Biology Guide. Great. That concludes topics 3.1 to 3.3, enzyme structure, enzyme catalysis and environmental impacts on enzyme function for AP Biology. Next is topic 3.4, cellular energy. Welcome to topic 3.4, cellular energy. By the end of this lesson, you will have a detailed understanding of the fundamental concepts of energy in living organisms, including its sources, utilization, and the significance of energy in sustaining life processes. There will be 4 parts in this lesson. Energy is the driving force behind all biological activities, enabling living organisms to grow, reproduce, and maintain their structures. In this comprehensive lesson, we will explore the intricate ways energy is obtained, stored, and utilized in living organisms, and how it plays a pivotal role in the sustenance of life. First, energy sources. 1. Sunlight. Solar energy is captured by plants through photosynthesis, where chlorophyll converts sunlight into chemical energy stored in the form of glucose. This process forms the foundation of the food chain. 2. Organic molecules. Heterotrophic organisms, including humans and animals, obtain energy by consuming organic molecules derived from plants and other organisms. These organic compounds are broken down through digestion, releasing energy during cellular respiration. Second, AP. Adenosine triphosphate is a nucleotide molecule that temporarily stores and transfers energy within cells. During cellular respiration, energy released from organic molecules is used to regenerate ATP from ADP and inorganic phosphate. AP hydrolysis releases energy, which is harnessed for various cellular activities, including muscle contraction, active transport, and biosynthesis processes. Third, importance of energy in living organisms. Energy is required for 3 main types of work: mechanical work, muscle contraction, transport work, active transport across cell membranes, and chemical work, synthesis of molecules. Energy is essential for building new cellular structures and repairing damaged tissues, ensuring the organism's growth and maintenance. Reproductive processes, such as gamete formation and embryo development, demand significant energy inputs. Fourth, energy efficiency and conservation. Organisms have evolved to be energy efficient, maximizing the use of available energy. Efficiency is crucial for survival, especially in environments with limited resources. Many organisms have developed energy conservation strategies, such as hibernation, estivation, and torpor, to survive periods of low energy availability. Great job! That concludes Topic 3.4, Cellular Energy, of the AP Biology exam. Next is Topic 3.5, Photosynthesis. If you found that helpful and want to listen to the rest of Unit 3, you can get the audiobook or ebook down below. Good luck on your exams! I look forward to helping you learn more about biology, human health, and the nature of life on Earth, and helping you succeed in your AP Biology course. Thanks for listening.